So we are done with Aspergillus. In the morphological classification, the third type is mouse. So the first one is yeast, second one is yeast like budding, and the third one is mouse. So in the mouse, we are done with one of the examples is Aspergillus. So the other one we see in this video is Zygomycetes. So Zygomycetes is a class, and the genera is Rhizopus, Mucor and Apsidia. So we will see Zygomycetes and Zygomycosis and Mucormycosis, and the differences between these three genera, Mucor, Rhizopus and Apsidia, and the lab diagnosis. So yeah, Zygomycetes, it is associated with Zygomycosis. So Zygomycosis includes Mucormycosis and Entomophthalmycosis. So Entomophthalmycosis is an infection of subcutaneous tissue and paranasal cyanosis. So yeah, Zygomycosis, these fungi are saprophytes of soil, manure and decaying vegetables. So Aspergillus, we said it's a ubiquitous, it presents everywhere, but these genera, these three fungi, Mucor, Rhizopus and Apsidia, are saprophytes of soil, manure and decaying vegetables. So these fungi are normally avirulent and are able to cause disease when there is general resistance is extremely low. So we said Aspergillus as a opportunistic pathogen, but these fungi are normally avirulent but only cause disease when there is general resistance is extremely low. So the primary infection usually seen in upper respiratory tract and nose, and the other one is rhino-orbitocerebral zygomycosis, also called as rhinocerebral disease. It is characterized by spreading lesions from nasal mucosa to turbinate bone, then to paranasal cyanosis, and then it goes to the orbit and then to the brain. So in the brain it causes necrosis and thrombosis and hemorrhage. So coming to the mucormycosis. So mucormycosis is caused by Mucor and Apsidia, and this mucormycosis is a invasive disease. So the firstly we will see in mucormycosis is pulmonary mucormycosis. This is a progressive severe pneumonia, and here these fungi spread hematogenously to other areas of the lung and also the other organs like brain. So yeah, the second one is gastrointestinal mucormycosis. This usually seen in malnutrition, uremia and diarrheal disease. So we will see the differences between Mucor, Rhizopus and Apsidia. So these differences are very important. It can be asked as a short note in your examination. So give a neat diagram and then you give a note about these three differences. Then you can score good marks. So we will see the three characteristic features in differences. Firstly is rhizoids, second is causes, and then sporangiophores. So rhizoids are present in Rhizopus and Apsidia, and it is absent in Mucor. So causes is mainly it causes, these three genera causes zygomycosis, and as I said, mucormycosis is caused by Mucor and Apsidia, and Rhizopus also causes otomycosis, whereas Apsidia causes keratitis also causes. These Apsidia causes zygomycosis, mucormycosis and keratitis. So yeah, coming to the lab diagnosis. So Zygomycetes is a class and these are genera. Mucor, Apsidia and Rhizopus. and it is associated with zygomycosis. So zygomycosis is a saprophyte, and the primary infection we see in upper respiratory tract and nose, and yeah, the other one is rhinocerebral disease. So coming to the mucormycosis, it is caused by mucor and absidia, and the firstly we have seen is pulmonary mucormycosis is a progressive severe pneumonia, and this fungi spread hematogeneously to other areas of the lung and to the brain. So gastrointestinal mucormycosis, it usually seen in malnutrition, uremia and diarrheal disease. Coming to the differences, mucor, rhizopus and absidia, and characteristic features I said rhizoids, causes and sporangiophores. So rhizoids are absent in mucor, whereas it is present in rhizopus and absidia, and the causes are mucor zygomycosis is caused by all these three genera, and mucormycosis is only caused by mucor and absidia, whereas rhizopus causes otomycosis and absidia also causes keratitis. So yeah, sporangiophores, it is present as branches in mucor, whereas in rhizopus in groups, whereas in absidia in between the rhizoids. So in mucor you can see the sporangiophore in groups, whereas in rhizoids, in mucor you can see the sporangiophores in branches, whereas in rhizopus you can see in groups these sporangiophores, and in absidia these sporangiophores are in between the rhizoids. Coming to the lab diagnosis, the specimen we take obviously scrapings from the lesions, and pus and sputum and nasal discharge. So coming to the direct microscopy, when we do KOH wet mount, we can see it shows a non-septate hyphae, and histological sections, hematoxylin and eosin stain, it shows the presence of hyphae. So culture we do is on Sebroid's dextrose agar without cycloheximide at 37 degrees centigrade. When we do lactophenol cotton blue, shows branched sporangiophores arising randomly along aerial mycelium. So in the case of mucor, rhizoids are absent, and rhizopus has rhizoids, and sporangiophore arise in groups directly above the rhizoids. So absidia has also rhizoids, but sporangiophores arise from aerial mycelium in between the rhizoids. So absidia has also rhizoids, but sporangiophores arise from the aerial mycelium in between the rhizoids. So lab diagnosis, the specimen we take is pus, nasal discharge, sputum and lesions. So direct microscopy, when we do KOH mount, shows non-septate hyphae, whereas histological sections, hematoxylin and eosin stain, shows the presence of hyphae. So when we do culture on Sebroid's dextrose agar without cycloheximide at 37 degrees centigrade, when we do lactophenol cotton blue, so we can see these characteristic features. So we are done with Zygomycetes. So we have studied about mucor, rhizopus and absidia, and the causes and differences and lab diagnosis. So I hope you all understand this what we are done with today. and I am making these videos for BSc MLT students and also the lab technicians. Even you all can revise. So yeah, in the next video I am coming up with the another example of mouse. Thank you.